Welcome. My name is Jelle Langendijk, and in this lecture I will talk about microscopy and staining methods. In this video we will explore how microorganisms can be visualized, the major types of microscopies used in microbiology, and several key staining techniques that help us understand microbial structure, function and activity. By the end of this lecture, you should have a good foundation for interpreting microscopic images and understanding why these methods are essential in environmental biotechnology and microbial ecology. Let's start with a brief look at history. The study of microorganisms began with Antony van Leeuwenhoek in the late 1600s. He built simple but remarkably powerful single-lens microscopes, some capable of magnifications of 200 to 300 times. With these microscopes, he was the first person to observe bacteria. His observations opened the door to modern microbiology, because for the first time scientists could directly see the microbial world rather than infer its existence. From the 1800s onward, microscopes improved rapidly and became widely used in scientific research. In the late 1800s, Hans Christian Gram developed the Gram stain to make bacteria more visible in lung tissue. It was later discovered that this method also differentiates the bacteria based on their cell wall structure. Around the mid-1900s, phase contrast and fluorescent microscopy were introduced. More recently, molecular imaging techniques and high-resolution systems have been developed, further expanding our ability to study microbial life. Before going into details, it's useful to understand the scale we're dealing with. Human-sized objects are visible to the naked eye. Cells and bacteria require a light microscope. Viruses, large biomolecules like DNA and atoms, require electron-based imaging technologies. Light microscopy bridges a huge range from a millimeter down to about 200 nanometers. Below that limit, the wavelength of the light becomes a physical barrier and we need electron microscopy. In this lecture, we'll focus on the techniques using light microscopy that are routinely used in microbiology labs, especially for studying mixed microbial communities. The most basic technique we use is bright field microscopy. A beam of white light is transmitted through the sample. Whatever absorbs or reflects the lights become visible. This method is simple, fast, inexpensive. Almost every microbiology lab has a bright field microscope. However, bright field has limitations. Many microbial cells are almost transparent because there are mostly water. As a result, unstained cells have a very low contrast and can be difficult to see. That is why bright field is often combined with stains, like the Gram stain, which we will discuss shortly. To overcome the issue of transparency, we can use a phase contrast microscope. This technique converts small differences in refractive index, basically differences in how much the cell bends the light, into visible differences in brightness. Because microorganisms contain internal structures with slightly different optical properties, phase contrast produces clear images without staining. This makes phase contrast ideal for viewing live cells, tracking motility, or observing changes over time without killing or fixing the sample. Phase contrast is extremely common in microbiology and is often the first choice when assessing cultures in their natural state. One of the most important classical staining techniques is the Gram stain, developed in 1884 by Hans Christian Gram. The goal is to differentiate bacteria based on their cell wall structure. Gram-positive bacteria have a thick layer of peptidoglycans and retain the crystal violet stain, appearing purple. Gram-negative bacteria have a thinner peptidoglycan layer and an outer membrane. They do not retain the crystal violet and appear pink after counterstaining. The stain is still a standard first step when classifying bacteria, diagnosing infections and characterizing isolates from environmental samples. For mixed microbial communities, the Gram stain gives only a coarse information, but is still a foundational method. Beyond bright field and phase contrast, a powerful approach is fluorescent microscopy. Fluorophores are molecules that absorb light at one wavelength and emit it at the other, typically a longer wavelength. Special filter sets control which wavelengths reach the sample and which wavelengths are captured by the camera or the observer. With fluorescent microscopy, we can label specific structures, proteins, or even specific microbial groups. This makes it possible to visualize phenomena that are invisible in traditional light microscopy. Where bright field and phase contrast use transmitted light, light that goes through the sample, fluorescent microscopy uses reflected light. The light has a different source and a different pathway. It comes out of the objective and reflects back through the objective. This works by using a filter cube in the path of the light. This filter cube only transmits light that excites the fluorophore in the sample. Light that is emitted from the fluorophore in the sample, that now has a different wavelength, is passed through the oculars or detector. In the next few slides we will highlight three important applications in environmental microbiology. We use live dead staining, body pie staining and fluorescence in situ hybridization. The live dead stain is used to assess membrane integrity, which is often used as a proxy for cell viability. It typically uses two components, calcine AM, which enters the cells and is converted into a bright green fluorescent molecule by intracellular enzymes. Only cells with an intact membrane and active metabolism will show this signal. The other component is a red dye that cannot pass through intact membranes. It only enters cells with damaged membranes, marking them as dead or compromised. By combining these two channels, we can quickly estimate the ratio of live to dead cells in the sample. This is particularly valuable when evaluating biofilm systems, environmental stress or reactor performance. The right image shows a single stained cells. The left image shows a very thin slice of stained aerobic granular sludge. Some bacteria accumulate polyhydroxyalkanoates, or PHA, as intracellular storage polymers. This is essentially a bacterial energy reserve like fat is to humans. The dye body pie binds strongly to hydrophobic structures and therefore stains PHA granules inside the cells. Here you can see in the left an image of bacteria with stained PHA granules inside the cell. In environmental biotechnology systems, such as aerobic granular sludge, PHA storage plays an important role in the carbon cycling and microbial competition. Using body pie staining, we can visualize where PHA is located inside these granules and how different microbial groups store carbon. On the right, you see a slice of AGS where the PHA is stained. It is mainly located on the outer layer of the granule. This method gives us insight into metabolic activity that would otherwise be difficult to detect. Now we move on to the most important tools for microbial ecology, FISH. FISH uses short fluorescently labeled DNA probes that bind to ribosomal RNA, primarily 16S rRNA. Because rRNA sequences vary between microbial groups, we can design probes that target specific taxa. After fixation and permeabilization of the sample, the probes hybridize to the ribosomes inside the cells. When we visualize the sample under the microscope, each probe fluoresces in a specific color, revealing the spatial distribution of microbial populations. For example, in the image shown here, red indicates Candidatus accumulibacter, green represents Candidatus competibacter. This allows us to study the organization of microbial communities with high spatial resolution. FISH is a very fast technique to monitor microbial communities. Within a single day, you can analyze the presence and abundance of specific targeted taxa. One of the key applications of FISH is the validation of other analytical techniques, such as omics. As shown on the right, omics data has predicted that only 25% of the community consisted of Accumulibacter, while FISH revealed that most of the cells were actually Accumulibacter. Therefore, FISH is a powerful tool to validate omic results. Recently, we developed a method to apply FISH to thin sections of AGS. This allows us to localize different microbial species within the biofilm structure. The image in the middle shows a full granule section, while the image on the left highlights the granule edge, where it becomes clear that different species occupy distinct layers within the granule. Let's wrap up with a short summary. Bright field microscopy is simple and fast, but it offers limited contrast for unstained cells. Phase contrast provides strong contrast without a need for staining and is excellent for observing live organisms. Gram staining remains a fundamental method to distinguish gram-positive from gram-negative bacteria. Fluorescence microscopy allows us to target specific structures or groups using fluorophores and specialized filter sets. Live dead staining helps us to assess membrane integrity and cell viability. BodyPy staining reveals intracellular PHA storage, and FISH enables precise identification and localization of microbial populations within complex communities. Together, these microscopy and staining methods are essential tools for linking microbial structure to function and for understanding how microbial ecosystems operate.